Hello, my name is Maxim Johnson, and today I will be presenting on the major differences in eukaryotic and prokaryotic gene regulation. So, this is a pretty short presentation. The rubric specified I only had to do two, so I'm only doing two, so it's really short. Yeah. I should say I chose. There are a lot of differences between eukaryotic and prokaryotic gene regulation. I chose the two that are kind of the most important, most distinguishing, and most, I guess, interesting overall is how I would put it. So difference one is location and timing. So in bacteria, prokaryotes, transcription and translation happen simultaneously. On this all happens inside the cytoplasm. This is, of course, because they don't have a nucleus, so there's not a defined structure for these processes to take place in. It just all kind of happens in a big muddle. And for eukaryotes, transcription happens in the nucleus, while translation happens in the cytoplasm. And of course, because translation is dependent on transcription, these processes don't happen at the same time. Transcription has to happen before translation can. So yeah, these processes occur in different areas and not at the same time. The second difference I chose was organization of, that is a typo. That's supposed to say genes. So in prokaryotes, genes are organized into operon. How is that pronounced? I can't remember. Operons? Operon? I want to say operons. And multiple genes can be transcribed into a single RNA strand. So, yeah, pretty self-explanatory. Multiple genes, same RNA strand, just works. This is called, well, I guess you could say, prokaryotes are polycystronic. That's like the term for it. Eukaryotes, on the other hand, are monocystronic, meaning each gene gets its own mRNA. So, on top of that, each gene is regulated independently. So it just tends to be a little bit more complicated for eukaryotes. Whereas with prokaryotes, you can kind of throw everything under an umbrella. With eukaryotes, everything has to have its own umbrella, which makes it a tiny bit more complicated. And as far as sources go, I used the Gen 3020 Lecture 14, Chapter 16 notes and slides, in addition to the Gen 3020 Lecture 15, Chapter 17 notes and slides. So those are just like the two lectures we had about prokaryotic versus eukaryotic. Uh, yeah, yeah, that's all.